when it comes to absorption, there are 5 different transporters. As I already told, it is dependent on transporters. So there are 5 different transporters who will be acting to absorb these amino acids which are digested from the proteins. Okay, what are they? First, transporter for basic amino acids. Okay, like for example, arginine, glycine. We have got a basic amino acid transporters. Next, for amino acid example, like for example, proline, we require specific transporter to transport this. Okay, proline. And also another amino acid, like for example, glycine. It helps absorption. Third, acidic amino acids. Yes, acidic amino acids. Acidic amino acids, like for example, glutamic acid, aspartic acid. They require specific transporters through which it can enter the cell. Fourth, like for example, if we have beta amino acids, all the amino acids are alpha. If we have beta, then there is a specific, like for example, beta alanine is absorbed only by beta transporter. Now, last one, what about neutral amino acids? Okay, neutral amino acids, like for example, alanine is there, leucine is there, isoleucine is there, valine is there, etc., etc. How they are absorbed? Neutral amino acids. The neutral amino acid absorption itself is very important exam question. Okay, we might expect a 3-mark question on this topic. That is the absorption of neutral amino acids. It is otherwise assisted. Okay, it is otherwise assisted by a tripeptide. You might guess that is the glutathione. The glutathione is a tripeptide. It's a tripeptide means it's again a protein which is containing 3 amino acids. What are the 3 amino acids? We have gamma glutamyl, cysteine, glycine. Okay, it's made up of glutamic acid, cysteine, and glycine. So it is made up of 3 peptides, tripeptide, gamma glutamyl, cysteine, glycine. So this molecule, in short form, we call it as GSH. Okay, this molecule has a role in absorption of neutral amino acids. So in the exam, they'll ask you for 3 marks, write the role of glutathione in absorption of amino acids. Means what the examiner is trying to ask you or make you to represent on an answer sheet, that how the glutathione helps in the absorption of neutral amino acids. Neutral amino acids. Now again, the glutathione is, as I told, it's a gamma glutamyl cysteine glycine. This cycle is also called as Mr. cycle. Okay, in the exam they might ask you, explain in detail about Mr. cycle. So what he is asking is to write about the neutral amino acid absorption, how it is absorbed by the intestine. Now, if we don't forget, where is the absorption? Absorption in the small intestine, because till it reaches the small intestine, the protein has become amino acid. In stomach, it is partially digested. In the duodenum, partially digested. Till it reaches the small intestine ileum, it becomes completely digested and becomes a single molecule single unit of protein. What is it called as? Amino acids. These amino acids are absorbed by 5 different transporters: basic, amino, acidic, beta. Okay, basic, amino, acidic, beta amino acid, and last is the neutral amino acid. So we'll see how this neutral amino acid is absorbed. This is called as Mr. cycle. Okay, this Mr. cycle is basically with the help of one molecule which I mentioned, glutathione. The amino acid, which amino acid? The neutral amino acid which is present in the lumen need to be absorbed into the cell. In simple, we can see it can be absorbed like this, but it cannot. Okay, it cannot be absorbed that way. Okay, it cannot be absorbed that way. So to assist the same, the glutathione comes into picture. What does glutathione does? The glutathione, with the help of the enzyme transferase, gamma glutamyl transferase, takes the neutral amino acid and forms glutamyl amino acid by releasing cystenyl glycine. Okay, the gamma glutamyl cystenyl glycine has converted into glutamyl amino acid. So amino acid has bind with the glutamic acid. Cystenyl glycine are split into glycine and cysteine. Now, what is our main function? We want free amino acid into the cell so that it can be absorbed and transported in the blood. Now, glutamyl amino acid is split to form oxoproline and free amino acid. That is cyclotransferase. Now, we want glutamine back because we want the cycle to continue. We don't want it to stop somewhere. We want it to recycle. If we want to recycle, we have to resynthesize the molecule. How we are resynthesizing the molecule? What we have separated? 3 compounds: glutamic acid, cysteine, and glycine. Glutamine is there, cysteine is there, glycine is there. Again, rejoin them. Okay, you mix them, you join them. What you are doing? The glutamate is converted into glutamyl cysteine, means glutamic acid combining with the cysteine. When you join something, definitely we require energy. So what we burn? We burn ATP. Okay, which enzyme is here? Synthetase. When we are talking about the use of ATP, the synthase, we call them as synthetase. If the joining is happening without ATP, we call it as enzyme synthase. Here we are joining the compounds with the help of the energy ATP, so we call it as synthetase. So if you see in the picture also, glutamyl cysteine synthetase. Now, what is left behind is glycine. We want to add the glycine now. So what we are doing? We are joining the glycine with the burning of energy. ATP is converted to ADP with the help of synthetase, that is glutathione synthetase, to synthesize the glutathione. So what is the final role? The amino acid, which was outside, is entering into the cell with the help of glutathione. The glutathione undergoes a cyclic manner where it is resynthesized. We are utilizing how many ATPs? If you see, 1 ATP, 2 ATP, and 3 ATP. Means what? For every 1 amino acid absorption, that is neutral amino acid absorption, we require 3 ATP plus 1 molecule of glutathione. Glutathione is recycled. Okay, it is recycled, so there's no problem with the glutathione. When it comes to ATP, yes, we are burning the energy. As I told, it's an active process. The absorption of amino acids is an active process. So we have completed the Mr. cycle where the amino acid is absorbed into the cell with utilization of the energy and a glutathione. Glutathione is a tripeptide, gamma glutamyl cystenyl glycine. Okay, please remember. And this is an important exam question which can be asked for 5 mark absorption, or only Mr. cycle can be a 3 mark question in the exam. So you should be able to write this detail in the exam with the structure. Okay, I hope till here we are very clear with the part of proteins.